Welcome to today's Explainer. We're embarking on a really incredible journey today, doing a comprehensive, multi-scale analysis of psychiatric disorders. We're going to zoom all the way from population-level behavior straight down into the genomic architecture inside our cells. For decades, the biological mechanisms behind mental illness have been kind of treated like a black box. But thanks to some absolutely massive advances in genomics and neurobiology, that box is finally wide open.
Here is our roadmap for today. We'll start with the foundations of psychiatric genetics, move into synaptic and network architecture, dive deep into chromatin regulation and epigenetics, and finally, look outward at future therapeutic horizons.
All right, let's jump right into Part 1, Foundations of Psychiatric Genetics at the Macro Level.
To really get a grip on this, we've got to reframe how we even think about psychiatric conditions. We can't just look at them as behavioral syndromes anymore. They are complex, heritable disorders of the central nervous system that fundamentally disrupt emotion, cognition, and motivation. And here's the wild part. The polygenic risk, or the heritability for these conditions, makes up anywhere from 20 to 90% of a person's vulnerability. 20 to 90%. That establishes a stark, undeniable biological reality from the literal moment we're born. To put that into perspective, autism spectrum disorders have concordance rates in identical twins of up to 90%. Schizophrenia? That sits at about a 50% concordance rate. The genetic weight here is just absolute.
But despite this immense genetic component, the field has historically faced a major hurdle. We've had a distinct lack of classic macroneuropathology or diagnostic biomarkers. Because of that gap, we're still diagnosing purely by clinical observations using manuals like the DSM-5. Well, there's a growing consensus now that these behavior-based classifications are completely missing the biological mark. They end up grouping together wildly different biological pathways just because they produce similar outward symptoms. It's like calling every car problem an engine issue just because the car won't start.
So this historical gap is driving a really necessary shift in our whole diagnostic framework. We're actively moving away from those subjective behavioral categories and towards something called the Research Domain Criteria, or RDOC. Instead of just slapping a depression label on someone, this approach classifies mental illness based on core behavioral abnormalities. Think things like a loss of reality or a decreased ability to experience pleasure. Then it maps those specific domains directly to the underlying brain circuitry. It's a total game changer.
Which brings us perfectly to Part 2, Synaptic and Network Architecture at the cellular level.
When we adjust our focus and look at the brain's wiring, modern imaging reveals some really specific interconnected limbic circuits that are physically altered in these disorders. We're talking about areas like the hippocampus, the amygdala, and the prefrontal cortex. In conditions like depression and addiction, these networks, along with the nucleus accumbens, actually have altered functional and structural connectivity. The physical communication highways of the brain are literally changing how they route traffic.
Now let's zoom in even closer on those circuits, right down to the actual gap between two communicating neurons, the synapse. This is where we find the real core of the issue. Genome-wide association studies show us that the physical architecture itself is where the functional disruption lies. We see it in the postsynaptic density of excitatory synapses. It's right there in the scaffolding proteins that hold the whole structure together. It's in the transcription factors regulating gene expression and heavily in voltaic.
And this introduces a super fascinating concept that is central to modern neurogenetics, pleiotropy. So a single variation in a gene, like that CAC11C we just mentioned, doesn't just cause one isolated disorder. No, that exact same mutation increases the risk for schizophrenia, for bipolar disorder, and even for severe neurodevelopmental syndromes like Timothy's syndrome, all at once. The same broken biological mechanism ripples out into vastly different behavioral symptoms based on a mix of other genetic and environmental factors.
Let's take it a step further. Part 3, Chromatin, Regulation, and Epigenetics at the Molecular Level.
We're zooming straight into the nucleus now to observe the genetic source code itself. DNA doesn't just float around freely in there. It's actually tightly wound around these sphere-like proteins called histones. This tightly wrapped structure is what we call chromatin. The absolutely crucial point here is that for any of those risk genes to actually be read and used by the cell, the chromatin has to be precisely unraveled and regulated. If the machinery that unwraps this DNA is broken, the brain's development goes entirely off script. And when you look at massive high-throughput sequencing studies, they've definitively categorized the genetic pathways involved in psychiatric vulnerability. They heavily point toward the exact cellular machinery that remodels and modifies this chromatin. Think about chromatin remodelers like ARID1B, CHD8, and ADNP. They physically restructure the DNA. Then you have lysine methyltransferases, like KM22C, adding methyl groups to histone proteins to modify epigenetic expression. When rare, highly penetrant mutations strike these specific genes, along with crucial ubiquitin ligases like RNF168 and TRP12, the risk for severe neurodevelopmental disorders like autism just skyrockets.
So you might be wondering, when and where does this genetic misfire actually happen? Well, when researchers map where these chromatin-modifying risk genes are most active, they consistently converge in a very specific place and time. They localize right in the glutamatergic neurons of the mid-fetal human cortex. Let that sink in. This tells us the biological foundation for many of these psychiatric vulnerabilities is being laid down long before we are even born, deep inside the developing fetal cortex.
Okay, let's zoom all the way back out. Part 4. Future Therapeutic Horizons, the Clinical Path.
Now that we've drilled down to the molecular origins, how does all this incredible neurobiological data translate into real-world personalized medicine? Well, the pathway to precision neuropsychiatry looks a bit like this. Step 1, identify a specific, highly penetrant genetic mutation in a patient. Step 2, develop nucleic acid targeting therapeutics. We're talking about cutting-edge tools here, like CRISPR for gene editing or antisense oligonucleotides, known as ASOs, which can block or degrade the faulty mutant RNA. And finally, Step 3, deliver these directly to the central nervous system to fundamentally correct the cellular misfire right at its source.
I really want to emphasize that these futuristic therapies are absolutely not science fiction. They are actively being pursued and developed right now. We already have incredible early success models using ASOs and spinal muscular atrophy, and today, researchers are applying those exact same targeted strategies to severe neurodevelopmental disorders like Angelman syndrome and Phelan-McDermott syndrome, which actually involves that SHANK3 deletion we touched on earlier. We're even seeing clinical trials using ASOs to target the mutant HTT RNA in Huntington's disease, and phase 2 trials targeting tau proteins in Alzheimer's. We are actively intervening at the genetic root.
So I want to leave you with a pretty massive question to chew on as we wrap up this explainer. If we are truly mapping these neural circuits, and if we're on the verge of deploying CRISPR and ASOs to directly rewrite faulty chromatin machinery, are we finally moving past the days of simply managing symptoms? As our magnification gets better and our genetic tools get sharper, we might literally be standing on the precipice of an era where we cure the biology of psychiatric disorders. Thank you so much for joining me on this deep dive today, and definitely keep your eyes on this rapidly advancing horizon.